I will discuss about something which is not directly hardcore plant pathology. Okay. So, thank you Vaibhav for inviting me to this beautiful students audience. The thing is that my lecture schedule was scheduled at 11:30. This is the first time I am seeing that he has given me a chance half an hour before. Okay. Very nice that whether half an hour after or before, that does not matter, because if you see the scale of time. So, 14 billion years ago, is there any concept of time? No. So, there was a very big event in the evolution of the entire universe. This was created due to a big bang, when there was no concept of time. Only after that the concept of time was started. So, half an hour earlier or after is nothing. So, anyway, so at that time of big bang, there was a very hostile environment in this entire universe. So, what was happening that time? Several storm, dust and different chemicals, they try to interact with each other, and during that process there was a first biologically active molecule created, and that was a RNA molecule. So, when that RNA molecule created, what happened then? This RNA molecule was catalytic RNA molecule. So, that can replicate itself, and then they produce several copies, and then they also interact with other things, many other chemicals, many other moieties joined together, and ultimately with this kind of biological material, the first cell, living cell originates. That was a first prokaryotic cell, that is not the nowadays bacterial prokaryotic cell, it was a very primitive prokaryotic cell. And then the evolution started. From that cell with one DNA or RNA molecule in it, it started replication, it started dividing, and then with the process of evolution, what happens actually, this unicellular single prokaryotic cell gives rise to different life forms in this earth. So, in that way all of we are related, are ancestor of one particular thing. Okay? So, why I am telling this? Because before going to tell about all these things, first you need to know that where we are at present. Our beloved mother nature created us, and it took billions of years time to evolve this kind of complex biological form in this earth. And we, the best creature of our mother nature, the humans, we are trying to now challenge with our mother nature. We are trying to solve the mystery by which the earth created us, the universe created us. We try to solve that mystery by just sitting on a computer. Okay? So, this is the story, this is the journey of a human civilization which started from a river valley, and now we are entering into a Silicon Valley. So, try to understand that how this evolution takes place and where we are standing. So, when we are the civilized creature of the mother nature, we are trying to ask question that from where we created, what is life, how the life evolved, how all the living beings in this earth are related, what is the element which carries all information and passed to the next generation. And to solve this question, solve these mysteries, people started thinking, the science started. And if you see the evolution of the science, during this era, the Darwin, Mendel, they started think about theory of evolution, how we evolved. Then Mendel gave the theory, principles of heredity. So, this time we considered the science that time as era of classical genetics, that how we evolved, how we are heredity is carried out. This got boosted up when there was happening. Yeah. So, there are after that there are several discoveries like nucleic acid discovery, DNA is the genetic material, structure of DNA was elucidated, and triplet codon was discovered, which started the era of molecular genetics. Okay? So, from the era of classical genetics, we slowly moved to era of molecular genetics. Now, in this time, what is actually people have started thinking that how this when discovery was made, the triplet codon. So, that time people started thinking about how from the DNA RNA is synthesized, and from RNA protein is synthesized, and this is called central dogma of life. So, this is the cellular biology review. So, what is happening actually in the field of science that time, that cellular biology, the classical genetics, molecular genetics, and this cellular biology, they are merging together. When they are merging together, a new branch of science actually evolved, and that is called era of cellular molecular biology. Okay? So, this era of cellular molecular biology got thrust when there was discoveries of several technology. So, what are those kind of technologies? Like sequencing of the DNA, polymerase chain reaction, cloning and recombinant DNA technology. So, when this era of technology comes, then it got boosted up, and again the history repeats. So, what happens that time? The classical genetics, molecular genetics, cell biology, and technological advancement, they merge together to give another era, the era of biotechnology. So, this technology, the sequencing technology, actually completely revolutionized the era of biotechnology, and with the discovery of sequencing technology initially by Maxim Gilbert or Sanger, then with different second generation, third generation, or different next generation sequencing technology like deep sequencing or nanopore sequencing or SMRT sequencing. So, with this different type of technology, sequencing technology, what is happening actually is generation of huge amount of sequence data. So, earlier a human genome sequencing project takes actually 20 years to decode the human genome, but now within one day we can do. So, it is so powerful, sequencing technology has come up. So, what happens? The cost of sequencing is going down, the cost of computing is going down. As a result, there is increased number of sequencing of different genomes. So, during this time, the late 90s, suddenly there was a boost because the new sequencing technologies came, and huge number of sequences were deposited in the gen bank or the repository of the sequences. So, they deposited huge number of sequences. So, when huge number of sequences deposited, it just look like this. Can you tell what is this? What is this? Anybody is having any idea? Any wild guess? Yes? Yes, it is not Harappans, it is hieroglyphs. Okay? In the Egypt, this language, these symbols were used to communicate with the people. So, it is a written language of that time. So, something is written. Are you able to read what is written? Why? Because you do not know what is written, what is the meaning of that thing. That is why you do not understand that thing. So, when sequencing technology generated huge amount of sequence data, it is just look like this kind of things. So, it is a just boring hieroglyphs, some A, T, G, C, nothing, combination of 4 letters, A, T, G, C. So, until and unless you are an expert of this language, you cannot decode what is written in this. Okay? And until and unless you are able to understand what is written in this, it is not possible to utilize this information, though we have lot of information, lot of data generated, but that data, that information cannot be converted into a knowledge until and unless you have the knowledge to read this data. Okay? So, the question is that, oh, it is not working properly. So, question is that when so much huge sequence information data generated, then the question is how different organisms are different. Who will find the function of all the proteins? Then what is the relationship between the sequence and the phenotypes? How these sequences are related? So, these questions are coming up. So, people initially used to think that okay, we are geneticists, we can only solve this mystery puzzle. So, it was only that time this kind of science was only restricted to the people who worked on genetics. So, then they realized no, it is not possible by only genetician to solve this problem. So, they think that okay, other scientists of the institute should work on that. That also failed, that one institute cannot solve. They now collaborate with different institute. When I was a master student, that time we do not have any commercial sequencing facilities, we did manual Sanger sequence, that time no softwares were available. I used to go to ICGEB, one international institute here, to do the molecular analysis, because only that institute could able to purchase the software. 25 lakhs that time was the cost of that software to analyze the result. But now almost all these softwares are freely available, because people understood that we cannot do by alone, not by a single institute. So, again the history repeats. The 4 different branch of science, biotechnology, information technology, computer science and statistics, they merge together to give rise to another new branch of science, that is called bioinformatics. So, with the help of this new branch of science, we are now started thinking that yes, we will able to solve what is involved or what is hidden in the ATGC, in the sequence information. But definitely as 4 different branch of science are involved in this subject, so it is not very easy to understand, definitely. Today's class, I am trying to tell you very lucid way what is there in the bioinformatics, what are the basic principles and practices which we follow on the basic aspects, because if you do not have this basic idea, then you cannot handle. So, you can stop me, you can ask me question if you are not able to understand, if I am going too high. So, the question is, what is bioinformatics? So, bioinformatics, what is bioinformatics? Before that, you need to know what bioinformatics is not, because many of the students, faculties, they do mistake. What mistake? They thought what bioinformatics is not, they thought that is bioinformatics. Okay? So, bioinformatics is not just using a computer to speed up biology. No. You have a computer, you have some software, you use that thing, you generate some result, that is not bioinformatics. Okay? It is not just applying computer algorithm to biology, and it is not just the accountant of the genomic data. So, these 3 things are not bioinformatics. Do not think that if you have a computer, if you have software, you can do bioinformatics. No. So, what is then bioinformatics? The first and most important thing is that creative use of the computer to define and solve biological puzzle. So, it is not only computer, the creative use of computer. It is not that computer will decide, it is you who have to decide what you need. Computer will not tell you what you need. Anything you give it to computer, anything you give data, you give it to software, software will generate some data. Okay? So, it is you who has to dictate computer, I need this kind of data for my work. So, it is not that computer will tell you. So, it is creative use of the computer to define and solve the biological puzzle, and computer is only help you to predict which needs to be tested at the bench. So, it is only a prediction machine. Thus, this bioinformatics only will able to predict something where you have to test whether that prediction is correct or not through your lab work. So, it is actually the marriage between the information science and biological science, this bioinformatics. So, just like the central dogma of biology, bioinformatics is also have a central dogma. What is that central dogma? If you have sequence similarity, suppose you have generated a sequence information of your any material, biological material, you have generated a sequence information. So, if that sequence information which you have generated, whether it is having any kind of similarity with any known sequence, okay, you have to find out the first thing that. And if it is having a sequence similarity, then definitely it is having a structural similarity. For example, if your sequence, which is we call them as query sequence means the sequence which we have generated now, and the match sequence means which is already known. So, if there is a similarity, then if this match sequence we know the structure is like this kind of things, for example, here I have given zinc finger structure, then your query sequence also predicted to have a zinc finger like structure. So, if you have sequence similarity, then you will have structural similarity. If you have structural similarity, then definitely you will have functional similarity. So, if the zinc finger domain of the match sequence is belong to a transcription factor, because this zinc finger binds to DNA. So, if it is having a DNA binding domain, this zinc finger, and it is belonging to a transcription factor. So, from there you can also predict that your DNA also have DNA binding activity, and this could also be a transcription factor. Okay. So, just by taking a sequence information, you can able to predict the function of a protein. Okay. So, this is the central dogma of bioinformatics. If you have a sequence, you can predict the function. Okay. So, bioinformatics has three major aspects. Being a pathologist, being a biological scientist, we do not bother about other two branches, because that is not in our domain. So, the first domain is that development and implementation of tools and databases. So, different tools and databases creation, this is the major domain of the computer scientist. Then development of new algorithms and statistical formulas for development of the software. So, again statisticians and computer scientist people, they are mainly involved in this kind of development of tools, softwares and other things. So, that is also again not our domain. The third domain is use of these tools to generate biological information and manage them. So, being a biologist, our role, our domain is this, that whatever software, whatever things that have been generated, we will definitely use them for analyzing our or solving our problem. Okay. So, whatever now I am going to talk about only about these things, that how we are able to solve different biological puzzles by using the different softwares. So, in this today's, because this is a very huge vast subject, only one part I am going to cover you, that is molecular phylogeny, principles and practice. So, when you generate a sequence information data, you have different, depending upon your organism, your biological material, you may generate sequence information depending upon your choice. You may have a small sequence information data of one gene, any one of gene of your organism or any biological material, you may have a only one gene sequence information. You may have a relatively big sequence information like bigger genes, smaller genome of viruses. So, that will come under this. You may have relatively big again, which is relatively bigger genome like bacteria, the entire genome of a bacteria. You may have whole complex genome like eukaryotic genomes, okay, like fungal genome. So, entire genome sequence data. So, depending upon your choice that which kind of data you are handling, you have to choose what type of softwares you will be using. This is the first thing, that whether you have a small gene or a whole genome, depending upon your sequence type, you have to choose what are the things you will be using. So, suppose if you have a small genome, small gene which is amplified by a PCR. So, you can generate sequence by just taking the primer sequence and generate only one read of the sequence. So, it will generate one read of the sequence like this. You may have a few more read if the genome inside of the your sequence is more, then what will happen? You can use two different primers from two ends and generate this kind of sequence information, and then there will be a kind of overlapping regions in this. So, what we have to do? we have to take this overlapping portion, we have to this sequence we have to take and you have to reverse complement it, because the entire sequence has to be from one end. It is not that from both the end you can take the sequence. So, you have to take this sequence, reverse complement it, so that this will come to this sequence, and then you have to join with them this earlier sequence and the overlapping region which is generated, you have to remove them and join them. So, this kind of things is called Contig assembly. Okay. So, if you have different read sequence, then you can join those sequences one by one to generate a bigger sequence. You remove the overlapping sequence, join the other reads. You may have cloned DNA. Suppose you have cloned your gene into a vector system. So, using a primer from the vector, you can amplify, you can sequence the entire gene. But in this case, what happens? You may have contamination with your vector sequence, because some sequence of the vector will also come. So, there are some softwares with which you can remove this vector contamination, and then you generate this only your gene sequence. So, this is called sequence purification. So, when you generate a sequence, first you purify your sequence, you either join them or remove the vector sequence, like that you first purify your sequence and come up only with your gene sequence. Majority of the students who are doing ITS sequencing or such kind of sequencing, they have this problem that there are many unwanted sequence they also take. So, only try to take only the portion which is of your interest, and remember that all the sequence should be of similar length for analysis. If the sequences are of different length, you cannot do proper analysis. That I will come later. So, for example, in case of funguses, we generally use ribosomal small and large subunit sequence, internal transcript spacer sequence, mitochondrial genes. There are several mitochondrial genes. So, this kind of different genes we can take out for the fungal genomes to do molecular analysis. Now, sequence of the large DNA, if you have a relatively large molecule, then we can fragment this DNA by mechanical shearing or by restriction digestion, and then we generate several small sequences. Then we overlap those sequences, we remove this overlap and ultimately join this sequence. So, in that way we can create a larger sequence data. For whole genome sequence, it will be like same, but in this case we add some adapter molecule to the fragments. So, that using this adapter sequence we can able to know that which portion is coming from which region, and this will generate three different things. The first one is called reads. Reads means only the initial sequence at a time when the sequence will be done, 200, 500, 600 nucleotide sequence, they are called reads of the sequence. So, these reads are aligned together so that the overlapping region can be removed, and then we join them together to form a contig. Okay. So, reads are aligned to make contigs, and then different contigs are joined, overlap together to form the scaffold, and then this scaffold, different scaffold joined together to form the whole genome. So, whole genome has to be divided into small parts, then lot of reads will be generated. These reads, related reads are assembled together to form contigs. Different contigs are joined together, overlapped, remove overlapping portion to form the scaffold. Different scaffold are joined together to form ultimately the whole genome sequence. Okay. So, in that way we generates the sequence. So, why I am telling all these things? Because this class actually I am going to talk all about this kind of basic concepts. Whoever is working on any sequence information, you may come across with reads, contigs, scaffold, this kind of terminologies. So, until and unless it is very clear to you what does it mean by reads, what does it mean by contigs, it will be, everything will be confusing. So, remember that reads are very small sequence information, reads are joined together to form contigs, contigs are joined together to form scaffold. Clear?
Sir, here reads
Yeah.
are like we are joining reads to form contig.
Yeah.
So here many overlapping fragment will be there.
Yes.
So like they are like to like for fragmentation or separation of the molecules.
Mm.
So on what basis they are solving? Like based on their complementarity or something?
Not actually when we will do, there is Contig assembly programs, what they will do, because this sequence information is generated based on this adapter sequence. Okay. So first you cut down the sequence into small pieces and then add some nucleotide sequence at both the end of the molecule, and then using that small sequence which you have added, complementary sequence of that you use as a primer to generate the sequence information. Okay, so you know that using this primer this many sequences have been generated. Now you all this sequence, the software what it will do, all the sequences they will do in the 5 prime to 3 prime orientation, 3 prime to 5 prime orientation, and they will try to merge them, that whether there is any overlapping sequence, whether same to same or means 5 prime to 3 prime and 3 prime to 5 prime. In that way they first convert one set of data 5 prime to 3 prime, and then they means it is called sliding. It is not directly your complementary things, it is sliding. One sequence is this, another sequence will slide along with that, and if it is finding the similar sequence, so that will be your overlapping sequence. So using the sliding the software will do, and then you will able to find out that where is overlap. So that portion you remove and join. So what the basic things I am telling, in the software you just upload the data, click on that, assemble contigs, it will do contig assembly. So for that you don't have to know that what is going on, but what is going on that I am telling, that what is the principle behind that. So until and unless you know principle behind that, then it will be very difficult that what things. Yes.
So in this way, information may be lost of removing the overlapping strands. We are cutting the overlapping strands, then joining. It may lack of some information. Information may be lost by removing this.
No, actually it is not that. From this region, for example, this sequence is also generated, this also is generated. So a little bit portion may be missing from here, but this will be having this portion. Okay. So it is not that only one sequence is generated from one read. Same area may be covered by several reads. So when if anybody has done any next generation sequencing or the, so they will generate several its frequency. You have to tell that how many frequency data you need for single region. In one region you may have several reads covering different lengths. Okay. So region is one, someone is starting from a little bit earlier, someone is going a little bit more. So there will be several reads. So when they will they will join, you will able to get all the entire sequence. It is very negligible that you will able to miss. So that is why there will be first a draft genome. So draft genome may be a near completion of 95%. So 5% genome may be lost. So you have to do again resequencing. So with the resequencing, the area which you are thinking that there is a gap, so that area you mainly cover, that if there is any gap in that, then you can incorporate that, and finally you come up to your complete genome. So at one go you will generate only a draft sequence, then you have to do resequencing to ultimately fulfill the a little bit portion, if anything is missed out, that you can fulfill.
Usually how much read depth will go for?
No, that depends upon how much money you have because you may have a 1 GB data, 1 TB data. So how many sequence reads you want, that depends upon how much money you will spend. So they will give you that many reads, that many frequencies of reads. So definitely if you have a 1 MB sequence read data and if you have a 1 GB sequence read data, definitely there will be huge difference. Okay, because you will cover majority of the read in small regions, so many reads will be you will get, then you will get more accurately the sequence of that. Okay.
What is the difference between read depth and coverage? Coverage, 10x coverage.
10x that coverage means, suppose this is the this is scaffold again, this is the whole genome. Suppose now few reads generated from here, this is from here like this, few area may be missed out. Okay. So, that tells you that how much coverage, whatever reads you have generated, that covers how much region of the genome. Okay. So, first you have to do a flow cytometry, this kind of things, to know what is the size of your genome. Okay. So, when you able to know that this is my size of my genome, then you will able to do the sequencing, and after sequence information, suppose you are getting that your genome size is 4 MB, and you are getting after sequence reads and other things, you are getting a sequence information of 3.89 MB. So, few reads definitely missed out. Okay. So, that means that much coverage, 90x coverage or 10x or whatever x coverage of the genome you have done. So, rest of the things you have to do by again resequencing of the things. Okay. So, this is the way how we generate the sequence information, and there are several softwares which will help you to do this Contig assembly. So, you can these softwares you can download, and you can see that how you can join different sequence reads to join the Contigs and able to generate the sequence information. But for whole genome sequencing or whole genome sequence analysis, using this some simple laptops or desktop computer, you cannot do. Okay. For that you need huge memory based computer and the open system like Sun Solaris and other things, so that you can analyze properly with that. Otherwise, if you start analyzing with your desktop computer, it will completely hang. Okay. It cannot handle that much volume of information. So, so this is a kind of whole genome sequence data. So, it is huge amount of sequence data. So,
Whether they have fixed any minimum amount of depth, minimum is when we are calculating QTL, we go for lot square of minimum 2.5 to 3. Is there any minimum value for that in this depth?
no, that depends upon what is your aim actually. Again, I am telling nothing is fixed. So, depending upon aim, you have to choose that what kind of sequence information you require. For example, we are doing a transcriptome data, we only wanted to know the gene sequence which are mainly the resistance gene kind of things which we need. So, for that we do not need all the entire genome sequence. So, if we are able to get the information on LBS, LLR kind of sequences, that is sufficient for us. So, we will not go for a whole genome or whole transcriptome, huge amount of depth or huge frequency data, we do not need. So, that depends upon what is your aim. If your main aim is to generate a whole genome sequence data, that is your thing, then definitely you have to go for very high depth sequence, then only you can cover the entire thing. But if you have only a little bit amount of things you wanted to know, for that you do not need that thing.
So, usually in paper we come across with N50 value for this sequencing. Then they use a stick like N50. What
So,
it means?
no, you just see that depends upon again different platforms which you are using. Okay. So, that value may be coming from Illumina. Okay. So, another company will give you another value. So, that is their means that company's prerogative, that what kind of things they have fixed up. Okay. So, when you give for sequencing, talk to that company that I need this kind of thing. So, their technical person will help you that how much means volume of sequence you need. So, that depends upon discussion, that depends upon that thing, that what kind of sequence data you need, they will able to provide you that this much depth you need. Okay. So, the main thing is that to find out the meaning of this information hidden in the sequence, we need to compare this sequence with the other sequence. So, if you have a sequence, you need to compare that with other sequence, then from where you will get that other sequence. The other sequence are available in different sequence databases. Okay. So, in this databases, sequence databases or biological databases, they are organized body of persistent data. They have three components essentially. If these three components are not there, we cannot call them as a sequence database. So, what are those database components? The first component is update component, second is query, third is retrieve component. So, update means you can update your sequence database. It is not closed. There are many database which is closed database, you cannot update that database, but all the sequence database are open database, you can update. So, if you have generated a sequence, you can update that sequence into the database. It is not that you have to send that sequence to somebody who will update that thing. Okay. Definitely there are some people who will check for the format and other things of your sequence, technical things, but you can update your sequence in the database. Okay. So, it is open. Then query component. So, in one sequence database, all the different sequence information, whether it is a human genome sequence, whether it is a rice genome sequence, whether it is a virus sequence, whether it is a fungal gene sequence, everything is deposited in one database. Now, it is very difficult for a person who is working on a rust fungus to find out where are the rust fungus sequences. Okay. So, for that the database should have a query component. Okay. So, using that query component, you can ask the database that I need this information. Okay. So, when you ask the database, then database will generate that information for you. Okay. Then okay, I want a rust fungus one particular gene sequence, beta tubulin gene sequence. So, you just give beta tubulin gene of rust fungus of that. So, it will generate the or it will collect all the sequence which is related to that, and it will show you. So, that is called query component. And third thing is retrieve component, so that you can take out that data from the database and you can analyze for your purpose. Okay. So, update component is there, query component is there, and then retrieval component is there. So, all this database should have. So, when we will do practical, that time I will show you where are those three different components in the database that you can use and you can take out your data. So, these sequence databases are mainly divided into two broad categories. One is sequence database, one is structural database. So, the sequence database are again of two types, nucleotide sequence database and protein sequence database, and the structural database is like protein database. So, here all the protein structured data are available. So, it is in the 3D structured data which is available in structured database in PDB. In nucleotide sequence, there are three major database all over the world. One is called NCBI, National Center for Biotechnology Information from USA, EMBL, European Molecular Biology Laboratory Database from Europe, and DNA Data Bank of Japan from Japan. So, these three are the major three databases throughout the world. So, any sequence information which you have to deposit any one of these sequence database. Now, you do not think that if I will deposit a sequence information in NCBI, whether I can able to get it from EMBL, or whatever sequence information is deposited already in the EMBL, can I access that thing if I will do from NCBI database? Yes, you can do. So, they have a internal collaboration among them. So, any sequence which is deposited in any of the database within 24 hours, that sequence will be accessible from any database. Okay. So, it is not worried that whether you are depositing your sequence in NCBI or EMBL or DDBJ, anywhere you can deposit the sequence. So, and it is the moral duty of all the researchers who are working with the sequence information. When you generate a sequence information, you have analyzed that and you understand that yes, it is okay, it is perfect sequence, then it is your duty to deposit that sequence into the database, because with that thing, the first one is the database will be updated, second one is your sequence will be visible to other, so that they can also utilize your sequence information for their analysis. Okay. So, this is the basic duty for all of you. So, besides this nucleotide, the giant 3 sequence databases, there are several specialized sequence database. Okay. Specialized sequence database means there is one sequence database for human genome, there is one sequence database maybe for Arabidopsis or rice genome or Magnaporthi genome, any such kind of things. So, you may have specialized sequence database only for that particular organism, that is also possible. But remember that whatever database you have specialized sequence, all those sequence also have been deposited in NCBI. So, until and unless that is deposited in this main 3 database, you cannot create a secondary sequence database. Okay. So, that has to be linked with these main 3 databases. Then there is protein sequence database like SwissProt or translated EMBL. So, if you have a protein sequence, you can deposit in these two kind of databases. So, these are the overview of what are the different types of databases which we use for analyzing or getting the sequence information of other sequence who have already been deposited, and you can utilize those sequence information for your analysis. So, this is the views means sprint view of this National Center for Biotechnology Information NCBI database. This NCBI database, which we also call them as GenBank, not GeneBank, GenBank. Then EMBL database, this is DDBJ database, then SwissProt and TreeMBL database, then PDB, this is the structured database. So, using this kind of online servers or tools, you can retrieve, update, do all those kind of things. Then these are some specialized database like Toraija Sativa specialized database. Then Magnaporthi sequence database, like that you have different type of databases. Now, the question is how you will deposit your sequence in the database. Okay, when you got a sequence, purify it, you know that yes, the sequence is correct, then you have to deposit the sequence in this database. So, for NCBI or GenBank, you have two options, one is called Bankit, another one is called Sequin, and for EMBL the option is called Webin Align. Okay. So, Bankit and Sequin is the format for GenBank. Suppose you have generated only one sequence information, okay, one gene or few genes, then you deposit through Bankit option. But if you have generated a cDNA library sequence or a whole genome sequence, so it is not only one gene, one portion. Okay. So, for that you have to use this kind of portal which is called Sequin. So, this both the things is available in the NCBI database. So, depending upon your choice. So, the first slides I told you, bioinformatics is not that having a computer internet facility. You have to choose, you have to direct the computer what you need creatively. So, if you have a one gene or few gene sequence, you should not use Sequin, you use Bankit. But if you have a cDNA library, you cannot do with the Bankit, because in cDNA library you may have 5000 cDNAs. So, individually 5000 gene sequence if you try to upload, it will take a month. Okay. So, depending upon your data, again I am telling you, you have to choose what kind of options what are available, you have to choose. So, that is why I am telling these things that you should able to understand at least after my class that okay, these are the, if I have this kind of data, these are options and I have to choose this kind of option. Okay. So, this is clear that how we deposit the sequence in the database. So, this is the Bankit option of the NCBI. So, you have to, you can read these things, you can just Google it and you can able to know that how to submit the sequence in the Bankit. This is the Sequin format, sequence portal in the NCBI through which you can deposit. This is the if you want to do in the genomic sequence or whole genome sequence, that is in the Webin Align. So, you can do like this. So, before submitting your sequence, you may heard this kind of terminology that we have to annotate my sequence. Okay. So, what do you mean by annotation? So, before submission of your sequence, you should annotate your sequence. So, annotation means it refers to the commentary or explanation of the information appended to DNA or protein sequence stored in the database. So, whatever sequence information has been stored in the database and which you are going to also store in the database, they should have some explanation, some appended information with that sequence, because if that is not there, nobody will understand what is this. Okay. So, what kind of annotation we can do? We can tell that what is the source of that thing, what is the country, organism, then protein sequence, predicted protein structure, domains of the protein, quaternary structure of the protein, protein function, common post-transcriptional modification and so on and so forth. So many things, whatever is available, you have to tell that suppose I have taken a beta tubulin gene sequence of rust fungus, then you have to first tell it is a beta tubulin gene of Puccinia striformis. Okay. So, if that is not there, nobody will understand what is this. Okay. So, that is the first information or first annotation, that you have to give a name, you have to identify what is this. Then if it is beta tubulin, from which region to which region is the sequence, because if this is a gene, definitely it will have some intron sequence, some exon sequence. So, you have to mention that in this entire region, this nucleotide to this nucleotide is exon, this nucleotide to this nucleotide is your intron. So, until and unless you tell that thing, whoever will be using that thing, nobody will understand what is this. Okay. And you have to tell from which country you have isolated, which plant you have isolated, like that you have to mention. So, this information which you are depositing, when you are depositing in your sequence, there will be these options will come. So, you have to mention as much as possible, so that if you give more annotated value to the sequence, your sequence will be cited much more, because we are seeing that several sequences have been deposited only giving the name, nothing is given. So, when you will try to analyze for your sequence, you will not able to take those sequences, because we do not know from which country it is there. So, how I will analyze, because after analysis you cannot do any inference, you cannot draw any inference from that, because nothing is appended on that. So, you do not do that mistakes, you should always try to put as much as possible annotation to your sequence, so that your sequence will be valuable more. Okay. So, you sequenced a gene or organism and deposited in the sequence in the database. So, up to this it is clear that you have sequenced a gene and you have deposited a sequence. Now, what is next? The next is data retrieval. Okay. So, data retrieval means collection of the data from the database. Okay. Again, I am particularly mentioning for the students, even if you can talk to your faculties, to your chairman, that sir, I would like to take out a sequence from database, or your chairman or any person or other friends may tell you, okay, download the sequence from the database. So, that is a very misconception. You cannot download a sequence from database, you can retrieve sequence information from the database. Okay. Do not tell after this class that I have downloaded 10 sequence from the database. Okay. Why? What is the difference between downloading and retrieval? Anybody tell? The downloading, suppose today I have given a mail to Vaibhav, and Vaibhav most probably has circulated to you a mail, attached some file. So, you download that software from that things. Okay. So, that is downloading. But when we are talking about a database, it is not downloading. Why? Suppose if we have a Word file, everybody has created Word file. That Word file may have 10 pages. Okay. If I will tell you, kindly download the 5th page of that file for me, can you be able to download the 5th page of that file? Is it possible? If you have created a 10-page Word document and if I will tell you download the 5th page of that Word file, you can download the entire Word file, but you cannot download only a page of that file. Clear? So, similarly, database is a file. Okay? And different datas are their pages. Okay? So, this data, your sequence information when you deposit, that is one page of that database or that file. So, one page of a file cannot be downloaded, that can only be retrieved. So, if I will tell you download the 5th page of the Word file, you cannot do. What you can do? You can copy that thing and paste it in another Word page and save it. Okay? So, similarly, when you try to take out a sequence information from the database, you have to retrieve it, you have to copy it, you have to paste it in a file, not that you can download it. clear? But this is a statistical terminologies. Many people do not understand. When we are student, even we are faculties, in the GenBank it was written that retrieve your sequence, and you can tick those sequence and you can retrieve. But there are several questions to the GenBank that I am not able to download my whatever sequence is there, I am not able to download, how to download. So, repeatedly GenBank people initially write mail that you cannot download, you can retrieve, and there is a retrieve button, you click that, it will be retrieved. So, people could not able to understand what is the retrieve and download. So, GenBank people got fed up. Now, they use the button as download, okay, because that kind of understanding concept is not clear. So, how many times you can tell people that it is not download, it is retrieve. So, it is better to use a download button, because it is a button whatever it is written. So, people very easily understand download, that is their mind, that download is the anything, if you take it from the internet, that is download, okay. But it is actually it is not download, though the button now it has been replaced as download button, but it is actually retrieval of the sequence, clear. So, collection of the data means retrieval of the sequence. Then after retrieval, actually what for we are retrieving the data from the database? There are several aspects for which we retrieve the data. The first thing is that we want to understand genomics or proteomics. Genomics means understanding interaction among genes, proteomics means understanding interaction among proteins, okay. Now, genomics may have different components like structural genomics. What is structural genomics? Like genome organization, ORF prediction or gene prediction, primer designing, all these kind of things comes under the structural genomics. Then comparative genomics. You want to compare your sequence with another sequence and predict what is the function. So, like ORF annotation, relationship, variability, phylogeny, molecular phylogeny, all these kind of things comes under comparative genomics. And then functional genomics. That means you want to predict the motif, function, blocks, domains, what is the function of the protein. So, you want to predict that thing, so that comes under functional genomics. So, if you have retrieved a sequence from the sequence database, then you can do all this kind of analysis for understanding the genomics or for understanding the proteomics, that is interaction of proteins with other molecules, drug designing, all this kind of aspect will come under proteomics, okay. So, you can do both this kind of aspect. Now, second term, another terminology comes, that is called data mining. So, data mining is generation of the information from the data in the database. So, in the database only the ATGC information is given, the data is given. So, from that data you have to generate information and knowledge, and this is called data mining, okay. So, there are several types of data mining. Some of this I have mentioned here, like molecular phylogeny, gene finding, primer designing, protein structure and function, promoter identification. So, all these kind of things comes under data mining, okay. So, mainly today I will talk about this molecular phylogeny, that how to understand the phylogenetic relationship between different sequences. So, up to this it is okay. Whatever I am telling, it is not going very up. If it is going up, you tell me, because subject is very difficult. I am trying to tell you in a very simple way. It is, the subject is not that much simple. So, okay, little bit of fun. What is this? Oh, how you understood? Anybody use this bullet? Really, it is surprising. Every time when I am showing this photo, everybody able to understand what is this. I am really surprised that whether you are doing research or you are doing some practice. So, it is bullet definitely. And what is this? Is pistol. And what is this? Again bullet. This bullet is thrown from that pistol. Okay. Now, what is this? Yes, it is the nozzle of the pistol. We are taking the photograph like that. So, it is inside the nozzle of the pistol how it looks like. So, it looks like some carved area. So, when you throw a bullet from a pistol, it will always go as a spiraling way, so that it will hit in a portion, but when it will come out, it is a big hole. So, always like that. That is why if you hit by a pistol, it is very difficult to survive, because it is not only going only in one direction, it is also spiraling. So, it creates a big hole when it will come out. Okay. So, this is the nozzle and there are grooves. So, when a pistol throws the bullet, it means creates some friction with this kind of beads or kind of grooves. As a result, there are several scar marks created in the bullet. Okay? It creates several scar mark in the bullet. So, what it looks like? So, it looks like this kind of scar mark in the bullet. So, it because it is getting friction with that nozzle bore, and then it throws. So, what happens? Suppose a bullet is thrown, it gets from this, it gets 2 scar mark. Second bullet is thrown from the same pistol, these 2 scar mark will be there, along with some more scar mark will be generated. Third bullet, when it will throw, so it will generate all those which is already there and some new, and like that. So, based on this kind of information, a detective can able to tell that whether all these bullets are thrown from the same pistol or not, because they analyze this kind of marks, and as suppose this bullet, as it is carries all those information from the earlier few information. So, they can correlate what is the ancestry of this bullet, from where it has been thrown. Now, you may ask question, why this kind of boring thing you are telling? Because suppose now this is the process of evolution. With this process, many things, many mutation, many things created in the individuals, and then the variation created in the next generation. But by taking some clue from this bullet and its nozzle, we can again find out that what are the particular sequences which is conserved across different individuals, so that we can track back from where it has been originated. Okay? So, from where it has been originated, if we are able to find out, then we can understand how these sequences are related. Just like how these bullets are related, whether they are throwing from the same pistol. So, we can understand whether from that same ancestry individual, whether all these sequences has been generated or not, based on some typical feature of the sequence which is conserved across all these sequences and related to the ancestral sequence. Okay? In that way, we can find out the relationship. So, the question is, what is this phylogenetics? Phylogenetics is the science of estimating and analyzing the evolutionary relationship. So, how this evolution takes place and how it is related, that is the science of phylogenetics. How it is done? It is done by analyzing the DNA sequence to help us to understand the phylogeny of the organism. What it is purpose? Phylogenetic relationship help us to understand the evolutionary history of an organism and how they are related to each other. So, now these things are coming, then that what is molecular phylogeny and what we want to know from this molecular phylogeny. So, this molecular phylogeny, the main aspect is that what is our sequence has any homology to any other sequences. These are the things we want to know from a sequence. When you have a sequence, definitely you should have a question that what are the things we want to know from that sequence. The first thing we want to know whether the sequence which I have generated, it is having any homology with any other sequence deposited in the database. Definitely this should be the first question. Then the question is, how much similarity is there between the sequence which I have generated and the sequence which has been already deposited in the database, whether any sequence similarity is there and how much sequence similarity is there. This will be the second question. The third question which we want to know from the sequence, that how our sequence is phylogenetically related to other sequence. So, these three are the important things which we want to know from a sequence which you have generated. Clear? So, again new terminologies are coming up. The first terminology, homology, second is similarity, third is phylogenetic relationship. So, what do you mean by homology?
Having same origin but diversified in different places.
Okay. Having same origin.
Homologous.
Anybody? Homology. Yes, ancestry. So, this is a state of gene or morphological characters that shares a common ancestry with a different gene or morphological character. So, if that is a state, that is a situation which shows us that there is a ancestral relationship with another sequence, then we call these two sequences as homologous sequence. Clear? Now, for molecular sequence data, it is taken to mean that two sequences or even two characters within a sequence are descendant from a common ancestor. So, if two sequence or a portion of a sequence or character of sequence is descendant from a common ancestor or not, that actually the purpose to know whether there are homologous sequence or not. But often this term homology is misused, rather abused by the students, by the faculties, because though you are telling that it is a state of gene or morphological characters, but still we do not able to understand what does it mean. So, what general thing we do mistake? We often, you can see any seminar anywhere, you can see your senior or your friends or your faculties, they are telling this term is frequently misused as a synonym for similar, because they are telling two sequences were 70% homologous, 80% homologous. Two sequences I have done, this sequence is showing 80% homology with another sequence. Many times people say like that, but this is completely wrong statement. Why? So, to understand that, just I would like to tell you a very nice quote from a biologist, Walter Fitch. He described the homology is just like pregnancy. So, you can either pregnant or not. Anybody can tell a woman is 20% pregnant, a woman is 80% pregnant? No. Why? Why you cannot tell? Because pregnancy is a state. It is not a quantifiable term. It is a qualitative term. Either a woman is pregnant or not. Yes, similarly, either two sequences are homologous or not. You cannot quantify homology. So, from after this class, never you will tell that two sequences are 80% homology. No, no, no. Homology, always when you will tell, remember this, homology is like pregnancy. So, we cannot tell 80% pregnant. Similarly, we cannot tell 80% homology. Okay? So, it is a qualitative term. So, homology cannot be measured, only we can say whether homology is there or not, but we can measure similarity. What we can measure? We can measure similarity. Homologous sequence must have similarity, but if there is similarity, we cannot say there is homology. Okay? So, if two sequences are homologous sequence, they must have high amount of similarity. But if there is similarity, we cannot tell these two sequences are homologous. We cannot tell. Okay? Understandable, the statement?
So, for similarity, we can say the woman is 3 months, they are 4 months pregnant.
The similarity is a measure of homology, definitely. And for example, what you are telling, it is a month, that we can tell. Similarly, that will tell you, yes, that is homologous sequence. So, homologous gene superfamily. So, whatever genes you will handle, anybody, they are belonging to homologous gene superfamily. Two major superfamily are there. One is called orthologous gene superfamily and one is called paralogous gene superfamily. What is orthologous gene? Orthologous genes mean, like example, DNA polymerase of goat, DNA polymerase of human. So they have similar kind of sequence, similar kind of function, but found in different taxa. Okay? So they called orthologous gene. So orthologous gene always means happens due to the result of lineage transfer. So during the process of evolution, when from one taxa to another taxa the gene flow happens, then the gene transfer from one lineage to another lineage, and that actually comes as a belongs to two different taxa. They are doing the similar function, but they happen to be in two different taxa. Okay? So this kind of sequence is called orthologous gene. Okay? So if you have a beta tubulin gene of your rust fungus, you may have a beta tubulin gene of a fusarium also. Okay? So that is called the homologous orthologous gene of the different organisms. Now paralogous gene. Oh, why it is happening? Paralogous genes like hemoglobin, myoglobin, they found in same taxa, same sequence, but different function. So their sequence is same, but one carries the oxygen to blood, another carries oxygen to muscle. Okay? So same genes, similar structure, everything, but function is different, found in same taxa. So in fungus also you will see the similar gene may be present in mitochondria, may be present in cytoplasm, they have two different functions. Okay? So this kind of paralogous gene arise due to result of a gene duplication. Okay? So during the process of evolution, two major process the evolution takes place. One is lineage transfer of the gene, one is duplication of the gene. Okay? So due to duplication of the gene, the paralogous gene superfamily created, and due to lineage transfer the orthologous gene superfamily created. So if you find in your fungus and everywhere, you will see several genes, MAP kinase, MAP kinase kinase, MAP kinase kinase kinase. So all are kinases, all are having similar kind of sequences. They are paralogous genes. Okay? But one gene is present in one taxa, same gene is present in another taxa, they are orthologous genes. So clear that what kind of sequence you will be handling actually. So till now what we have understood, that if you have a sequence, your main target is to find out homology. Clear? But you cannot tell percent homology. So indirectly what you have to do, you have to understand what is whether there is a similarity or identity. Again two terms come, similarity, identity. Okay? Don't tell indiscriminately, don't abuse these terminologies, because everything has a different meaning. So until you will understand this basic concept, you cannot tell my sequence is 80% similar or my sequence is 80% identical. What kind of things you will tell? So what do you mean by similarity or identity? So I am going to give you an example. So this is one sequence, this is another sequence. You are trying to compare these two sequences. So you can see that these three nucleotides, A, T and here A, in both the things it is called identical sequence. Okay? So if you are trying to do analysis based on this identical match sequence, then you have to tell these two sequences have this percentage of sequence identity. Okay? So that time you should not tell this is the percent similarity, you will tell only sequence identity. Now besides this identical match, you may have this kind of match. A pyrimidine is replaced by a pyrimidine, a purine is replaced by a purine. Okay? In the two sequence. they are not identical match, but they, as per their base composition, we can tell they are similar match. So, similar match is actually including the identical match will also be there definitely, but along with that the similar match, if you do the similarity percentage, then they will calculate these things also. So, in the software you will have all these options, whether you need a similarity percentage or identity percentage. You will think that both are same, what is the difference? Similarity जो भी है, identity भी वही है, but no. Similarity includes both identical sequence as well as the sequence which may not be identical, but as the chemical composition they may be identical. Okay. So, if you are analyzing through identity based matrix, then you mention it is this much percent identity is there. If you do similarity based matrix, then you tell this much percent similarity is there. So, depending upon again your choice of option, you have to tell whether two sequences are how many percent similar or how many percent identical. Okay. So, it is not that random use of a software, random use of something and generate some result, tell something. So, software will generate something, you will tell some other things, and the reader who will read, they will understand something else. Okay. So, now after this kind of class, now you should not do these mistakes. Okay. No further calling of data downloading, no further calling of percent homology, no further calling of arbitrarily this much percent similarity or identity. Clear? Now, another thing is there, which is this kind of sequence. A pyrimidine is replaced by a purine. Okay. This kind of sequence is called as mismatch sequence. Okay. So, mismatch sequence is there. Now, suppose when somebody will tell you that try to compare two things, how you compare? Compare two things, any two things. Morphology, then you can compare. But in case of sequence, when I will tell you compare the two sequence, there is no morphology. So, for sequence, how we will compare? How we are comparing here two sequence? Same position. If it is one first nucleotide, we are comparing with another sequence of first nucleotide. Okay. So, here as there is no morphology, we cannot tell that इसका face ऐसे है, इसका face ऐसे है. We cannot tell. But here we can tell based on the position. The first nucleotide, what is there in the one sequence, first nucleotide, what is there in another sequence. So, this is kind of things we do, that we do analysis through positional similarity or identity. We compare the two sequence based on positional similarity or identity. Okay? And this positional similarity or identity is called alignment. Okay? So, from the first slide I am talking about that you have to do sequence alignment and then you do. So, what is sequence alignment? Sequence alignment is a process, is an algorithm by which we can compare two sequences based on their positional similarity or identity. Okay. So, this sequence alignment is an computational hypothesis which identifies positional similarity or identity between bases or amino acids. And this alignment is of two types. One is called global alignment and another one is called local alignment. Okay. One is called global alignment, second one is called local alignment. So, what is global alignment and local alignment? Suppose it is your query sequence. This query sequence is matching with a portion of a target sequence which is deposited. Okay. So, if you analyze through local alignment, it will only show the stretches of region which is showing maximum sequence identity. If maximum sequence identity region is there, then they will only search on that aspect that this region is showing only identity with the target. But in case of global alignment, if the target sequence is big and your sequence may be small, but it will try to analyze from the first nucleotide of the target sequence with the first nucleotide of your sequence. Okay? So, this is called global alignment. Understand? Any problem? So, in case of local alignment, suppose you have 500 nucleotide sequence you have generated. Out of this 500 nucleotide, when you will try to analyze, you will see 300 nucleotide sequence is showing very close identity with any other sequence. So, the software will only analyze that 300 nucleotide which is showing very high sequence identity. It will not take up the other things. But if you give the software command that I want a global alignment, then it will try to take up the entire sequence and try to, there may be not that much matching, but few matchings are there, they will show that also. So, depending upon again your choice that whether you need a local alignment or global alignment, you can ask the software to do. Now, you may ask question, what for we will do local alignment, what for I will do global alignment. So, when you have a generated a sequence, you have to analyze your sequence initially to know whether really this sequence having any worthy sequence, whether it is really means showing anything with any known thing. So, that time you have to do a very quick analysis, a quick analysis you have to do, then you only ask the software that I need only a local alignment to first give us a glimpse that whether it is having any identity with any sequence, that may be a small stretch of identity, but whether it is having anything. Okay? And when you find out yes, it is having some identity, then retrieve those sequence from the database and then do a global alignment to clearly understand how much sequence identity is there. Local alignment cannot tell you how much sequence identity, because it is doing only a stretch of sequence, not the entire sequence. So, when I will do practical, that time I will show you this local alignment, global alignment, all those kind of things. So, but you understand, means try to remember that there are two types of analysis, local alignment, global alignment. Local alignment is done to very quickly generate the information about the homologous sequence deposited in the database, and through local alignment we only able to find out the stretches of sequence which is showing high degree of similarity or identity, but we cannot able to tell how much percentage of sequence identity is there. That is not possible through local alignment. Only we can identify, yes, these are the sequence with which my sequence is showing identity, not more than that. So, how to align? If you see one alignment, just one example I am giving with few sequence. So, this is one sequence GAAATTC and this is another sequence GATTC, only this much. So, there are two nucleotides shorter than this sequence. Now, if you try to align these two sequences just by position wise, keeping those things, then what we are generating? There is 2 match, 3 mismatch and 2 gaps. If this 2 match, this 3 mismatch, and there are 2 gaps. But these 2 gap, if you can put here and here, then you can see it has increased the match. Okay? Mismatch decreased to 2, gap is same 2. If you put this also here, then you can see the match is increased to 5, mismatch 0. So, the same sequence we can align by different ways, different position changing and other things. The main motto of the sequence alignment is how to minimize the mismatches, how to minimize the mismatches and increase the matches. So, if there is much more matches, then the alignment has great value, otherwise it will not have value. But putting this kind of gaps which is not actually present in the sequence. In the sequence there cannot be any gap. Okay? But as this sequence 2 nucleotide shorter, so we can play with these 2 nucleotide thinking that there could be any mutation in early evolution days, and we can place this kind of gaps in the sequence. That is our will. Nature will not tell you that whether there is a gap or not, because we want to maximize the match sequence. So, we introduce such kind of gap to generate a good match alignment. Okay? So, this alignment, when we get this kind of things, we are doing some unnatural things, that putting some gaps in the sequence which is not allowed. That is why if you put a gap, this kind of gap is called open gap, and then put another gap following the first gap, then it is called extended gap. So, when you are giving this kind of open gap and extended gap, you have to give some penalty. Okay? So, what happens, software what it is doing, it will do this kind of different types of permutation and combination of this kind of alignment, and it generates a score value based on this match sequence. For example, here match is 2, it is giving a value of 2, here match is 3, value is score value is 3, here value is 5, score value is 5, like that. Okay? But it is score value is 5, but you are putting 2 gaps. So, what it will do, that if you are suppose putting one gap, 0.5 marks will be deducted. If you put another extended gap, 0.25 marks will be deducted, like that. So, though value is 5, but as you are putting some gaps, you have to give some penalty, and they are called open gap penalty or extend gap penalty. So, if you put more open gap penalty and extend gap penalty, you have to give more value of, you have to increase your penalty value. So, your score value will be reduced. As a result, what will happen, that it sometime it may be possible that this match may be better than this match, because you are putting too much gaps and adjusting the alignment. So, your total score value may be reduced. Okay. So, software actually will do these things. You just see, with the 6-7 nucleotides, you have to do lot of this kind of permutation combination. If it is 1000 nucleotide sequence, and if you are analyzing 10 different sequence of that, it is not possible by human being to do manually. So, here is the need of the algorithm, and this algorithm, so how to do, if the sequence is large, we need some progressive alignment, and this progressive or dynamic algorithm of programming is done to generate score. So, software will do this kind of things and generate against each pair of algorithm, it generates some score value, okay, and E value. So, two things it will generate, one is score value and one is E value. So, when we will do practical, that time you will be able to see that when after an analysis, the software will have generated a score and E value. So, more score value means more similarity, and E values is actually the probability of not matching the sequence with other sequence. So, again it is a statistical term. Statistics always try to show the nose in other way. They will not directly show it nose is here, it will show the nose is here. Similarly, E value means the probability of not matching your sequence with other sequence. So, if E value is the logarithm value. So, if E value is 10 to the power minus 3 means it is 0.001. Okay. So, this 0.001 is the probability of not matching your sequence with other sequence. That means, probability of matching is 99.999. Okay. So, directly it will not tell that probability of matching what is, it is rather tell you probability of not matching your sequence. So, the E value more means E value more means E value more means your sequence has less similarity, E value less means your sequence has more sequence similarity. Okay? So, you will be able to see this kind of score and e value generated by the software, from which you can infer that these are the sequence with which your sequence shows high level of sequence similarity or identity. But again, just by this kind of alignment, local alignment, you cannot tell how much. It will tell these are the sequence which will show high level of similarity. So, these are the sequence alignment tool we generally use: BLAST, FASTA, BLEACH, BEAUTY, CLUSTAL W, MEGA. So, among this we will show you this BLAST, the MEGA, CLUSTAL W. These are the things in the practical we will show you how to analyze this tool to do sequence alignment. Okay? So, just for theoretical purpose, what is BLAST? Remember, this is not bomb blast, because I have seen in many paper, many students, their thesis seminar, I have blasted the sequence with this. You have not blasted. BLAST is a program name. It is not a operation you are doing, okay, that you are blasting. So, BLAST is a program. So, you have to tell I have analyzed my sequence with BLAST. Okay? BLAST is an algorithm, is a program. You have to tell that I have analyzed my sequence with BLAST algorithm, not that I have blasted the sequence with other sequence. Never, because these are the common mistakes students are doing. Okay? So, what is BLAST? The full form of BLAST is Basic Local Alignment Search Tool. Okay? That is why this BLAST comes, Basic Local Alignment Search Tool. So, you see that it is a local alignment search. It will not do a global alignment. It will only do a local alignment and tell you only, yes, from the database these are the sequences which will show you high degree of sequence identity, but how much it will not tell. So, BLAST is the algorithm used by a family of 5 programs that will align your query sequence against the sequence in a molecular database. So, it will align this algorithm. This gives a statistical methods are applied to judge the significance of the matches, that how much significant is the matches. And alignments are reported in order of significance as estimated by the applied statistics. So, they will do this and use applied statistics and generate some score value and e value. Based on the score value and e value, it generates the significance, that how much significantly it is matching, and the first match it will show is the most significant match, next the next significant match, like that. Okay? So, BLAST is comprising of this different program. The first one is called BLAST-N. This compares a nucleotide query sequence against a nucleotide query database. So, if you have a nucleotide sequence, you use BLAST-N. Again I am telling you, it is you who have to tell the software what you need. If you have a nucleotide sequence, you want to know that whether my sequence is showing any similarity at the nucleotide level with any other gene, then you have to do BLAST-N. If you have a protein sequence, amino acid sequence, you want to know whether that protein sequence is showing similarity with any other amino acid protein sequence, then you have to do BLAST-P. Now, if you have a nucleotide sequence, you want to know that whether my nucleotide sequence codes for any protein, okay? Whether the nucleotide sequence which I have generated, whether it is generating any protein and it is showing any sequence match with any protein sequence. You have not done any protein sequence, only you have nucleotide sequence. But if you want to ask the database, the software, that whether the sequence which I have, the DNA sequence which I have, whether it codes for any protein, for that you have to do BLAST-X. Okay? What is happening? And last one is T-BLAST-N. So, if you have a protein sequence, amino acid sequence, you want to know that whether the protein sequence or which gene codes this protein sequence. You have amino acid sequence, you do not know which gene codes this protein. So, you want to know which gene may code this protein sequence. So, that time you have amino acid sequence, you want to go back to the reverse orientation, to know which gene is coding this protein. So, that time you have to do T blast N. Okay. So, these options are given in the software, in the online tool. You have to choose according to your choice, it is not that arbitrary thing. You have to tell the software, yes, I need this much, this kind of analysis, then only software will do that. Otherwise, you give anything, you put any aspect, it will generate anything, because software is a black box. It can generate any information, whether it will be required by you or not. So, after generating data, you may not able to infer anything, because you have not given the proper input, and that is depending upon you how you will use effectively the computer. So, a blast analysis looks like that, that if you give a query sequence like that, and if you ask this online server to tell that what are the sequence with which your sequence is showing similarity, you paste this sequence and do a blast analysis, it will generate like this and generate these are the different accession number. And remember that whenever you deposit a sequence in the database will give you a accession number of the sequence. So, it is a kind of patent. So, that sequence other people cannot just copy and deposit again, because you have already deposited this sequence and you got a accession number from the database. So, these are the accession number, these are the sequence description, this is the score value, total score, then query coverage, then E value. So, this query coverage means whether it is taking the entire sequence or which portion of the sequence it is showing for the similarity analysis. So, like that it will tell E value and all these things, and it will generate this kind of score. So, this practical we will do in the afternoon, that how if we have a sequence, I have already circulated that mail, most probably Vaibhav. You download those softwares, you install those software, and there will be 2 notepad file. So, those 2 are the test sequence which we will do today afternoon. Okay. So, what we know from BLAST? We know from whether the sequence shares similarity with query sequence, what are the sequences, and it helps us to retrieve those sequences. So, BLAST analysis only tells you that what are the sequence which is showing similarity with your sequence, and it will allow you to retrieve those sequence from the database. It cannot quantify the sequence similarity, because it is a local alignment. As it is local alignment, it cannot quantify, because it is only taking one portion, rest of the portion it is not taking count, and cannot tell us about the relationship between all those sequence. As it is again taking only a few stretch of sequence, it will not able to tell you that whether it is having any phylogenetic relationship or not. Why I am telling these things? Just see the example. So, I have suppose given a sequence, it is the score value 488, and this is the E value, and this is the identities. So, out of 373, it is showing similarity 346. So, 373 sequence, it is showing 346 nucleotide which is showing similarity. Based on that, the percentage is coming 92%. Okay. So, this is the sequence, it is the query sequence, it is the subject sequence. So, it is showing somewhere, it is not showing, it is showing gap. So, these are the gap sequence are there. So, what is happening? So, this is the entire length of sequence, and your sequence is matching like this. Okay. Some gaps will be there, and your sequence is matching like this. Now, coming to the same sequence, if you scroll down, you will get this kind of sequence where score is less. Earlier score is very high, here score is less, and here it is showing 84 nucleotide and 77. So, out of 84 it is showing 77. So, in the entire stretch of 370, it is only taking 84 stretch which is showing similarity, rest of the things is not showing similarity. Okay. So, it is like that this is the entire stretch of sequence, only this stretch of sequence is showing similarity, not other things. But within this stretch of sequence, if it is 84, it is showing 77, that means again it is 91. Okay. So, earlier it was 92%, here it is showing 91%, but in this 91% is only based on this region, not based on the entire thing. So, if it would take this entire thing, definitely it will be less than 91%, okay, because this sequence is not showing similarity, that is why the software has not taken these things. Now, if you do a global alignment from this end to this end, definitely the sequence identity will be much more lesser, and the total sequence identity will not be 91%, it will be much more lesser. Okay. So, if you are doing only local alignment, though local alignment will also generate a score value, a percentage sequence identity, but that identity we are not going to consider, because that is taking only a portion of sequence, not the entire sequence. So, that is why you have to take out those sequence again and do a global alignment, then only we will able to know whether it is exactly showing how much sequence identity. Clear up to this? Yes.
We have two strands of the DNA.
Yes.
While depositing this one, whether we deposit both the strands or
Depend upon your gene is there, if it is having a gene,
if we consider the whole genome,
then whole genome there is no problem, only one sequence strand you have to deposit,
one single strand.
only sequence strand. But if the gene, some gene is there in one strand, some gene is there in another strand, you have to mention all this annotation, you have to mention that from this nucleotide to this nucleotide one gene is there, but that gene is in 3' to 5' direction. Okay. So, when you deposit this sequence, you have to tell all those things in the sequence annotation, that how many genes are there, which position which genes are there, whether it is in the positive sense, whether it is in the negative sense, which region in that gene is the exon, which region of the gene is the intron. So, all this kind of information you have to append along with your sequence when depositing. Okay. So,
Go for the use of blast, whether it will take the if it is in the other strand which we are not depositing.
yes, everything, because whenever you search a sequence, that sequence may be in a negative direction, means ulta direction, and your sequence is deposited in positive direction, it will tell you, yes, it is also showing you, but there you will see this kind of things, plus plus. That means the sequence which you are giving, that is also in the same orientation as it is deposited. If it is not showing this, it will show plus minus. That means the sequence which you have sending, that is in the minus, but the sequence which is deposited, that is always in the plus. Okay. So, it will tell, because when the software will analyze, it will analyze in both the sequence. You may be deposited in only one sequence strand, but when you try to analyze, it will analyze with both the strand of the sequence. So, that software will do quickly, okay, and it will generate that whether it is plus plus or whether it is plus minus. Okay. That is not a problem. So, this kind of, due to this kind of local region alignment, we call them as local alignment, not the global alignment. So, to do global alignment, we do multiple global alignment using some other software, which we will show you also. So, here what we will do, we generate quick pairwise alignment. So, pairwise alignment, suppose you have retrieved 10 sequences, now you have to do a pairwise alignment. Pairwise means from first sequence, second sequence, first and third, first and fourth, similarly second and third, second and fourth, like all this pairwise combination of the 10 sequence, the software will generate a pairwise score value. And then from this pairwise score value, they calculate a distance matrix. So, you can see here that with this 24 and these things, the two sequence, they generate this kind of score value, and then they generate 1 and 2, 1 and 3, 1 and 4, these are the things, individual pairwise they generate this kind of score value. So, they generate score value, and then they generate a distance matrix and try to correlate that which two sequence pair are having less distance. Less distance means they are close. So, they take those two sequence as a group and join with the next sequence which is coming closer to that group. Clear or not? Not clear. Suppose if you have this kind of sequence 1 and 2, 1 and 3, all those kind of things, they are 99, 92 something. So, this is 99, that means 1 and 2 is score is 99, they are so much similar. So, 1 and 2 they make a group, then with this group they will try to find out which one sequence will be coming closer. So, with that they will join the next one. So, in that way they are trying to build up a tree. The first two sequence they put these two sequence, join them, and then next sequence which is coming closer they will join with that. So, in that way they are trying to build up the tree. So, this tree is called guide tree. So, based on this guide tree then they will do progressive alignment following this guide tree. What is progressive alignment? They will take two sequence alignment and then take the third one, but they will not change the whatever gaps has been created already for doing the alignment of the two sequence, they will not change the gap again. They will try to align the third sequence with keeping this already established gap, because you have to do a progressive alignment. So, in that way all the sequence one by one they will take and they will do a multiple alignment like that, and then from this guide tree then finally a multiple alignment global alignment file will be created from the first nucleotide to the last nucleotide. Okay. So, there may be some sequence may have big gaps and other things because their sequence is shorter or something. So, you cannot see that some sequence will be very much close sequence, some are very much diverse sequence, but they will create a kind of matrix or alignment file, and based on this alignment file then you can generate a phylogenetic tree. So, this is the kind of alignment file. So, using the software BioEdit, which you will do also today afternoon, this software. And from this software you can also generate this kind of sequence identity matrix. Okay. Now, only after the multiple global alignment you are able to generate a sequence identity matrix, which will tell you with your sequence what is the percentage of identity other sequence is having. Okay. Before that you cannot tell. So, this kind of percentage sequence identity you can generate, and then you from this percent sequence identity again you can generate a relationship tree, okay, that with your sequence how the other sequences are coming. Okay. So, so this kind of tree is called phylogenetic tree. So, what is a phylogenetic tree? This looks like this. This phylogenetic tree is having different components. The first component, it is called a root. It is called a root of a tree, from where the tree started. Then this is a region where the other things are coming out. So, this is called a node. So, these nodes represent taxonomic units, okay, and these are called branches. So, these branches represent relationship. Okay. That if these branches are coming together within a node, they are having close relationship. Then the branches of this and this, they are having distant relationship. So, the branches which are having close relationship, they are grouping together. Branches which are having distant relationship, they are grouping differently. Okay. And these are called sister taxa. So, the two branches in a clade, this is called clade, they are called sister taxa of a clade. Okay. And this is the length which indicates the genetic distance. So, this is the length of the branch which is called genetic distance. Okay. Now, this is clear? This terminologies, first you need to clear the terminologies, because often you will face this kind of terminologies in your work or in the research paper or anywhere. And there is another things in this kind of L form, where these taxonomic units are there, nodes are there, you will see some values, numbers. They are called bootstrap value. this tells us the significance of these two branches to hold together. Okay? Significance of these two branches to hold together in one clade. Huh? Then the significance is less. If it is more, then the significance or you can tell the probability of these two branches to hold together in one group is true, strong. I am coming, all these points I am coming now. You are not feeling bored? Something new you are learning? Okay. So, the branching pattern of a tree is called topology, that how these trees are branched, it is called topology, and the representation of the relationship through line is called dendrogram. The relationship is called dendrogram. Many people may have seen phylogenetic tree or dendrogram, both are same. Phylogenetic tree is only the dendrogram which represents the phylogenetic relationship. And the topology is how you are seeing or viewing the tree, that is called the branching pattern is called topology. Now, this dendrogram is divided into two parts or two types of dendrogram is possible. One is called phylogram, another is called cladogram. So, in the software you will see all this kind of options are there, depending upon again your choice. You have to decide which kind of phylogenetic tree you need. Okay. So, there is a phylogram option, there will be a cladogram option, and both phylogram and cladogram could be represented by a rooted tree or unrooted tree. Okay. So, again 4 new terminologies: phylogram, cladogram, rooted tree, unrooted tree. So, what is phylogram? Phylogram is a phylogenetic tree that indicates the relationship between the taxa and also conveys a sense of time or rate of evolution. The temporal aspect of the phylogram is missing from a cladogram. Clear? So, in the phylogram we not only group them, we also convey a sense of time. Okay? The sense of time means the time with respect to its evolution, how they are evolved, those sequences. So, if your analysis is related to that kind of conveying time, then you use phylogram. If it is not there in your study, do not use phylogram. I have seen many students paper anything unknowingly they are whatever software will generate, they are giving and showing them. But software by default it will show you a phylogram, but if it is not required by you, you do not use phylogram. So, how the phylogram looks like? A phylogram looks like this. Okay. So, here you will see this is one clade which is going, this is one clade which is stopping here, this is another clade, so many branching and stopping here. So, these ends are not at one position, these ends are in different positions. Okay. So, this kind of tree is called phylogram. Always in the phylogram you will see a kind of bar here, a small bar, a small measurement which will tell you this much distance indicates something, and that indicates the time actually. Okay. So, that you have to tell that if it is 1 centimeter means 1 million years. Okay. Suppose you want to analyze that how the humans have developed from different stages, different skeletons or whatever you have got and different sequence you have, then you can do by taking that time that after this many million years the humans from the chimpanzee it started to bipedal, then this and that like that. Okay. So, that kind of analysis if you want to do, then you have to use this kind of phylogram. Now, in this phylogram, what are this branching pattern means actually? This means, so this group as it is going further, so this is the group which is evolutionary more advanced group, evolutionary more advanced group. The next group evolutionary more conserved, because this length is conveying the time, okay? This length is conveying the time. So, here if the time is 1 million years for example, so after 2-3 million years they started developing in newer forms, but here they are not dividing into new forms, only after so many million years they come out with 2 different organisms. So, they are evolutionary much more conserved group. And in this, the last one, as there are so many branches, so these are evolutionary much more diverse group, because much more changes rapidly happening. Okay? So, if your data, if you try to analyze through this kind of orientation, that I have to tell that this is the group which is evolutionary much more diverse group or conserved group or this kind of data, and I have to convey sense of time also, then you use this kind of phylogram. If your data is not related to that, you do not show phylogram. Okay? For that you have to show cladogram. Cladogram is the depiction of the hypothesized branching in order to number of sequence. Cladogram do not give any indication of temporal change, but only phylogram does. Okay? So, it is only taxonomic grouping. Cladogram is only grouping. So, how the cladogram to look like? It looks like this. So, all the things will come at one place. So, there is no meaning of this branch length, because you are not conveying any time. It is only grouping. So, this is one group, this is another group, like that. This may come from Africa, this may come from Asia, like that you can group. So, if it is only for taxonomic grouping, you can use this. So, this type of cladogram is called rectangular cladogram or phenogram. So, there is another type of cladogram which is called slanted cladogram. So, the cladogram may be rectangular cladogram or may be slanted cladogram. Okay? So, this slanted cladogram, again you can use the slanted cladogram to analyze some evolutionary aspect, but without any time thing. So, what are the things you can do? You can study like convergence, divergence and parallelism in this, and here when we want to do grouping for the taxonomic study you can do, and in this case you can do when you want to study convergence, divergence or parallelism in evolutionary studies. So, if you want to study how these sequences are, whether it will converge in future, whether it will diverge in future, or whether it is a parallel evolution. So, that kind of analysis you can do through this slanted cladogram. So, what do you mean by this convergence, divergence and parallelism? So, for example, after analysis you are able to find out that these 2 sequences in future it may converge again, and these are parallelism, they are evolving without interfering each other. A rice, a human, a fungus, they are parallelly evolving, they do not need each other. So, parallel evolution or convergence evolution or this kind of divergence, that after certain time they diverged out. Okay? So, how they will diverge out? So, if you are trying to analyze your sequence based on this type of analysis, you try to interpret your result based on this kind of analysis, then you use slanted cladogram. So, again I am telling you, I have seen to many students that for the beautification purpose they try to give a different types of tree, circular, goal, this branching, this, that. Do not do, science is not for this kind of beautification. You have to use the software for a creative things, for actually solving the answer, whatever things you try to give answer or whatever things you try to interpret, the data will support that. It is not that अरे उन्होंने किया है कितना गोल गोल, कितना सुंदर किया, हम भी करना है गोल गोल. अरे गोल गोल क्यों करना है? If your data doesn't support that thing, why you will do? Okay. So depending upon your data, you have to choose whether you choose phylogram, slanted cladogram, rectangular cladogram, what kind of cladogram or phylogram you want, that depends upon you. Okay. So when you will do actually in the practical, that time I will show you all these options where it will be available. So you have to choose that what kind of things you, otherwise software will generate any data you give, software will generate anything. Okay. So you have to tell software what I need. So then another two terms, rooted dendrogram and unrooted dendrogram. Okay. So what is rooted dendrogram? So this is rooted dendrogram. It is having a root, and in this case we don't have any root. Okay. So we don't have any root. Root means in this case we assume that all the tags are derived from a common ancestor. If you assume that all the tags are derived from a common ancestor, then we use this. If we assume that the tags are not derived from a common ancestor, then we use this kind of unrooted dendrogram. Suppose you try to analyze the evolution of the life form in the earth, you take bacterial sequence, you take human sequence, you take rice sequence, you take any other sequence. Okay. Definitely they are not having from same thing. So in that case we use this kind of tree. But if you have a Puxchinia striformis beta-tubulin gene of different isolates, definitely they will originated from Puxchinia striformis. Okay. So in that case we should not use this, we only use this. But I have seen many students, if you are already anybody has handled the sequence and generate such kind of tree, you may have done a goal type of tree, idhar, udhar, idhar. So this is a unrooted tree. If it is rooted, then it should have a clear cut idea, otherwise it is everything is goal, 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 goal. Okay. So that is not required, because if you are working on one organism and different isolate sequence you are trying to compare, do you think that this different isolate of same fungus will originate from different fungus? No. So you should not use such kind of unrooted tree, only you have to use the rooted tree. Okay. Now how we develop this tree? What is the method of development of tree? So there are two basic way we can generate the tree. One is called character based method, another one is called distance based method. So the character based method is analyzing the tree based on a character of a sequence, one nucleotide sequence they are choosing, character based method. So this character based method, suppose here it is T, in another sequence it is G. So it will generate a score of 1 and then it will calculate a matrix and generate a tree. Okay. So from each score value it will generate matrix and it will generate tree. Now this kind of character based method, when we generally we can use, suppose if your gene sequence is very, very similar, means almost all the nucleotides will be similar, only few nucleotides will be different from one isolate to another isolate. You are handling a rust fungus, 50 isolates you collected. So definitely if it is rust fungus and 50 isolates, they will have the similar sequence only few nucleotides will be varying, it is not that huge amount of gene sequence will be varied. So if in that condition, we will definitely assume that only few nucleotides will be variable, and in that condition you have to use character based method. Okay. Another condition, you have taken a rust fungus, a blast fungus, a fusarium fungus, and you try to understand in one gene whether there is variation. Definitely in that case there will be lot of variation. Okay. So in that case only few nucleotides may be similar. Clear? So in one case maximum nucleotides will be similar, only few nucleotide will be dissimilar. In another case maximum nucleotide will be dissimilar, only few nucleotides may be similar. See, in both these cases we have to use this character based method, where only few nucleotide variation we can detect. Okay. So that is called character based method, and another method is called distance based method. Here we are not calculating a character based, but it is a distance based. Why? For example, we have taken this 3 sequence, there is a mismatch between these, so we generate a score this. We took another sequence where again it is T. Now this T, it may be possible that it is that, so the first and third one score is 0, but it may be possible that earlier it was G, due to back mutation it again come to T, okay, it may happens. So one generation passed, it again converted into T, okay, but we are not calculating that generation distance, okay. So directly telling that kind of things is impossible here. So for that we generally used a distance based matrix. So a distance based, so score is 0, but genetical distance is 1, okay. So this genetical distance based matrix we create, and this distance based matrix when we create, then we generate the tree, okay. So these two type of basic tree we generate, either character based tree or distance based tree. So in the character based approach we have the algorithm which is known as maximum parsimony or minimum evolution or maximum likelihood. These are the two algorithms you will often encounter in the softwares, in the papers, that the phylogenetic tree was constructed based on maximum parsimony algorithm or maximum likelihood algorithm or neighborhood joining algorithm. Like these are the different algorithms, based on these the trees have been created. So these algorithms are actually either based on character based approach or based on distance based approach, okay. So these two algorithms, maximum parsimony and likelihood, they are based on character based approach. So maximum parsimony, when you will use this, when the sequences are quite similar, very similar sequence, only few nucleotide we are assuming they are different, and there are small number of sequences you are handling, that time you use maximum parsimony. If you are handling same different isolate of same fungus, then you use maximum parsimony. If you are using, when the sequences are more diverse sequence, different gene sequence you are trying to, of different organism you are trying to analyze, you are expecting that there will be much more diverse sequence. So in that case your assumption is only few nucleotide will be similar, maximum sequence are dissimilar. So you use maximum likelihood. Likelihood means maximum how many sequences will be same. Here maximum parsimony or minimum evolution. So minimum how many sequences will be different, and here maximum how many sequences will be same, okay. That is why it is called maximum likelihood, this is maximum parsimony. These two are the character based method. And then the example I am trying to show you, for example human, rabbit and chicken, these three organisms I am trying to do a multiple alignment, and after that we observe that in this there is one nucleotide difference, in this there are 3 nucleotide difference, here also 3 nucleotide difference, okay. So based on, so this is the score value 1, 3, 3. So based on that we generate a matrix between human rabbit 1, human chicken 3, rabbit chicken 3. So this is called matrix, this is the data, this is the score value, and this is the matrix. And from this matrix now we generate a tree. How we generate a tree? So based on these things, which one is having less value, that means which one come closer, human and rabbit, because here the value is less, that means among so many nucleotides only one nucleotide is mismatch, so it is much more closer. So human and rabbit we put them together in a group, okay. Then with this group chicken and rabbit, they chicken is coming, and it is coming in this way that with chicken and human we have 3 score value. So this 0.5 and 1, 1.5 and this 1, so chicken will come like this, and rabbit this 0.5, 1 and 1.5 again 3, okay. So in this way we generate a phylogenetic tree based on character based method. Clear that how the phylogenetic trees are created by the software. So, they take this score value, generate this matrix and then generate this tree in this way. Now, coming to the distance based approach. So, what is distance based approach? So, this is one aspect that is called neighbor joining method or neighborhood method. So, neighbor joining method is a distance based approach where we take the sequence and create a distance matrix. So, it is a kind of distance matrix. So, we are taking spinach, rice, mosquito, monkey, human. These are the sequence we try to analyze, we try to do a phylogenetic grouping, that how spinach, rice, mosquito, monkey and human are related, okay, and this is the distance matrix. So, in the distance matrix what you are seeing, that these two are lowest value, distance value is low. So, the matrix may be different type, this is called distance matrix, there may be a substitution matrix, may be a similarity matrix. So, in case of similarity matrix we will take the highest value, in case of distance matrix we will take the lowest value. Lowest value means they are closely related and similarity matrix means they are highly related, that is why we will take the highest value. So, in this distance matrix this 3 and 3 is there. So, it is between human and monkey. So, between human and monkey there is lowest distance, that means they are closely related. So, what we will do, these two things we will take. So, in the first step we create a group human, monkey, we will create a group human, monkey as a group. Okay. Then what software will do? Software will try to understand with this group which one will come closer, rice, spinach or mosquito. Okay. So, they will create next cycle, they will create now another matrix keeping monkey and human as a group. So, monkey and human with these things, the distance matrix created, and this comes as mosquito with monkey human comes closer. So, what it will do? It will join this monkey human group with the mosquito. So, now it will create mosquito monkey human a group. So, the tree is developing. Now, in the next cycle it will try to understand which one is come closer, spinach or rice. So, it will do another round of distance matrix creation. So, now it is showing the with mosquito monkey human the distance is not that much related to any other, rather between rice and spinach it is showing lowest distance, that means they are forming a group. So, this grouping is not grouped with this. Okay. So, what will happen? It will form a group like that. So, rice and spinach will form another group. So, this is one group, this is another group. Okay. So, if we take only up to this, we call this kind of situation as monophyletic phylogenetic tree. That means only one phylum is there, only one group, and under that there are several. But here we are seeing another group. So, it is polyphyletic origin. So, there are two different origin of evolution, one is evolving this group, another origin will evolve this group. Now, we can combine them together to make the entire picture. So, what will happen in the entire picture? Now, you can join them together to make this entire phylogenetic tree. So, in this phylogenetic tree, it is a polyphyletic phylogenetic tree which is having two major clade. One clade belongs to monkey, human, mouse group, and another phylogenetic tree, another origin is spinach and rice. Okay. In these two, again in this clade there are subclade and subgroups which are belong to this. So, in that way you can analyze your data. Okay. So, in that way the phylogenetic trees are created, and you have to analyze your data on that, how many clades are created, in one clade how many groups are there or how many sequences are there, like that you have to now analyze. Now, you can correlate. Now, here is the importance of the annotation. Now, suppose if your these are your fungus sequence and these are forming one group, and from the annotation data you find that all these sequence of that fungus comes from Asia and this one comes from Africa. So, you can make these two groups, that one is Asia origin group, one is Africa origin group. Then in the Asia you will find another subgroup, then try to find out whether host. So, these are coming from wheat, these are coming from other host. So, you can make them based on the host. So, in that way whatever thing sequence is there, try to correlate with the annotated sequence, try to correlate with its biological function, and then you will be able to properly analyze how these sequences are related. Yes.
Actually, if we represent two phyla in a same telegram, then whether they have the ability to converge, like spinach and mosquito, whether they can converge.
Yes. So, whether they can really come together, whether they really come together, this question automatically comes. Na? To understand this, we need to understand what is the significance of this grouping, whether they really can come together or not. So, here again I am trying to give one very nice analogy with this. Suppose when you are now going to get married or somebody may be married. So, before marry, generally in our country we used to do some horoscope matching and all other kind of things. What for? What for? To try to predict that शादी का बाद ये दो एक साथ रहेगा क्या नहीं, है ना, इसलिए तो. So that is the same thing we have to do कि दो ग्रुप तो बना दिया, इसका साथ इसको रहना है, लेकिन वो इधर वो दो चीज एक साथ रह पाएगा क्या नहीं. That is the thing we have to now understand. So that horoscope matching we have to now do, that whether the grouping तो हम जोर जबरदस्ती करके बोल दिए इसका साथ इसका ग्रुप होगा, लेकिन मान लो एक लड़का एक लड़की को ग्रुप बना दिया, फिर और एक लड़का आ गया, वो लड़की उसके साथ चला गया, ये अकेला हो गया, तो वो देखना है कि फिर फर्दर कोई सीक्वेंस उसमें आएगा, then whether it will separate out or whether it will be grouping remains same. So how much is the attraction to each other, so that they will always hold together and they will not divide, even some more sequence will also come, they will not separate out. Okay? So that kind of horoscope matching we have to do, then only the significance of the tree is there, otherwise it is not there. So that significance of the tree is called the bootstrapping. Okay? So the last topic is bootstrapping of this tree, to understand whether these grouping whatever software is generating, they holds true with many other sequences. So bootstrapping is a statistical method which judges the confidence of the tree, judges the confidence of the branch, yes, this they will stay together. So there are different methods the software will do, but the basic thing is that, for example, this tree sequence you have taken sequence A, B, C, and first you generate this kind of tree sequence A and B in one group and C in this, and there is 70%, means it is a bootstrap value of 70%, it will come like that, in the they will do this again alignment with different replications. How? In the first step, if this is 0 to 9 nucleotide, for example, then they will interchange this nucleotide randomly. So 1, after this 5, then 6, then 2, like that, they interchange. In the second replication, they will again change this nucleotide sequences within this, and every time they will generate again a tree. Same sequence will be there, only they change some position of the sequences, and again they will do. So here you will see again it will come A, B closer, C alag, A, C here closer, B alag, here A, B again C. So out of this 3 replication, 2 are coming same, 1 is different. Okay? So ultimately a consensus tree will be generated, which will tell that if we will do 100 times, for example, here only 3 times I am showing, if we will do 100 times, 70 times there is a probability that even the sequence will be changing, they will be staying together. So this is called bootstrapping. So many times, many replication, many permutation and combination in the sequence the software will do, and then they will do the bootstrapping, and out of 100, out of 1000. So, that depends upon again you have to tell the software whether we will means calculate the bootstrapping of 100 times replication, 500 times replication or 1000 times replication. So, more amount of time it will give, more time it will take by the software to analyze definitely. So, that many times it will do replication, means different permutation combination, and again construct phylogenetic tree, and again tell you that out of 1000 times how many times after changing the sequence also, how many times this sequence tree which have been generated first, they are holding together, the same branch holding together. Okay? They will test this significance, and out of this, if it is value is, for example, it is 90, means 90% value is there, that means definitely it is out of 100, 90% or 99% it is showing a very strong identity. So, in that case the phylogenetic tree which we have developed, mosquito, monkey, spinach, rice and other things, there may be a very high bootstrap value between mosquito, in between monkey and human. It will be very much lesser with monkey human group and mosquito. It will be much more lesser in case of rice, spinach with that group. But again with rice and spinach it may be the significance or bootstrap value may be higher definitely. Okay. So, depending upon that kind of value we can infer that whether our phylogenetic tree is holding true or not, that grouping is really true or not. Okay. So, there should not be any confusion. So, choose a set of related sequence, the first aspect. Now, coming to the conclusion that if you have a sequence generated, curated, deposited and got the accession number, do the analysis, take out set of sequence from the gen bank and then try to start with the analysis, obtain a multiple alignment of those sequences and then ask the question that whether there is any strong similarity or not. If there is strong similarity, yes, strong similarity is there, because through this multiple alignment you can develop the identity matrix, you will be able to know that how much similarity is there among all the sequences. If there is high amount of sequence similarity, then if it is yes, then you go for maximum parsimony method, then do the bootstrapping and generate the phylogenetic tree. Now, if it is no, that strong sequence similarity is not there among your sequence. So, strong sequence similarity means 90%, 95%, 98% similarity among all the sequences it is there, not two sequences. With all sequences it is 95, 98. So, it is very strong sequence similarity. If it is there, you go for maximum parsimony. If it is not, then ask the question whether there is clearly recognizable sequence similarity, that is 70%, 80% is there, or even it is more less than that. Okay. So, whether any recognizable sequence similarity is there. If it is there, yes, then do a distance matrix, okay, neighborhood joining, and generate the bootstrapping and phylogenetic tree. If there is no clear recognizable sequence similarity, if it is 20%, 10% like that sequence similarity, then again go for another character based method, that is maximum likelihood based method, and then do bootstrapping and design the phylogenetic tree. So, depending upon your data set, what sequence you are having, what sequence you are retrieving from the database, you have to tell the software whether you need a maximum parsimony based tree, whether you need a maximum likelihood based tree, or whether you need a neighborhood joining tree. Do not do it arbitrarily, do not depend upon the software to guide you that you develop a neighborhood joining tree. If your sequence is if you have this kind of clear idea that what is my sequence data, then you direct the software that I need this kind of tree. Okay. Do not allow the software to guide you. So, from the first slide I am started to telling you, do not guide it by the software, you guide the software what to do. Clear? So, bioinformatics, we are started with the intelligent creature, and we are trying to understand everything by sitting on the computer. But really many times it is not holding true, there are a lot of limitations, it can only predict, but actual thing you have to test. It can give many times erratic result. A different subject are interlinked, so it is very difficult to understand in those things, and it may be possible to make relationship and models, but in reality they may not be appropriate. So, who will tell the last answer, our beloved mother nature or we, by sitting on a computer? Let us see. Thank you. Any question? कुछ तो बोलो. Understood? Very difficult subject. Clear? So, it is little bit clear with the theory. When you will do practical, I think many of the things you will understand much more better way. When you will do the practical in the afternoon, bring some laptops, make some groups, charge it